Good morning and welcome to Channel 23 News. Always original, never a phenocopy. In today's traffic report, I'm Lindsay Moss-Taylor.
And I'm Maya Raghuvandan.
Joining us tonight will be special correspondent Joanna Mooney.
And a very special guest, Takeshi Arashiro, Esquire, DDS. Today we bring to you a heartbreaking story of actin and myosin.
Breaking news!
What is it?
PDGF regulated RAB4-dependent recycling of alpha V beta 3 integrin from early endosomes is necessary for cell adhesion and spreading!
This is just incredible. Well, it looks like actin and myosin will have to wait.
Oh no. It's been postulated that the regulation of integrin vesicular traffic facilitates cell migration by internalizing integrins at the rear of the cell and transporting them forward within vesicles for exocytosis at the leading edge to form new contacts with the extracellular matrix. The RAB family of GTPases control key targeting events in the endo and exocytic pathway. Therefore, these GTPases may be involved in the regulation of cell matrix contact assembly.
Well, actually, it turns out that growth factor-regulated RAB4-dependent recycling of alpha V beta 3 integrin from early endosomes to the plasma membrane is a critical upstream event in the assembly of cell matrix contacts. They wanted to investigate the mechanism by which platelet-derived growth factor, PDGF, regulates integrin trafficking and what effects does it have on the cell.
Previously on the traffic report, we've seen that PDGF treatment increased levels of alpha V beta 3 integrins at the plasma membrane. In this breaking report, this group has developed assays to measure integrin endocytosis and recycling to determine if PDGF regulation could account for these changes in alpha V beta 3 surface levels.
Now to go to our expert, Takeshi Arashiro, DDS Esquire, to tell us more about how these alpha V beta 3 integrins localize within the cell. Takeshi Arashiro, over to you.
Hello, this is Takeshi, and I'll explain this figure showing immunofluorescent images of localization of alpha V beta 3 integrin after 15 minutes and 30 minutes of internalization. Integrin staining is shown in green, RAB protein staining is shown in red, co-localization is shown in yellow. With 15 minutes incubation, integrin is co-localized with RAB4 in early endosome. With 30 minutes incubation, integrin is co-localized with RAB11 in perinuclear compartment. RAB4-mediated endocytic recycling is a faster process and a shorter route than the RAB11-mediated recycling pathway. Back to the studio.
Thank you, Takeshi, for all the insights.
Now to our special correspondent Joanna Mooney, who's reporting from the scene. Joanna, can you hear us? How's it going?
Hi Lindsay, Maya. I'm here on the scene at the poster session with a report that can only be seen here on Channel 23. To answer the question of how PDGF affects the recycling of integrins, researchers pulse-labeled surface integrins with biotin, allowed them to be internalized, and measured the amount of recycling with addition of PDGF and without. What they found after 15 minutes is that a greater percentage of alpha V beta 3 integrin was recycled to the plasma membrane with addition of PDGF. On the other hand, alpha 5 beta 1 recycling was not affected with or without addition of PDGF. When the reaction was allowed to go for half an hour, however, there was no significant difference between the recycling of either integrin with or without PDGF. This links the recycling of alpha V beta 3, but not alpha 5 beta 1 to the fast loop recycling pathway regulated by RAB4. Just to confirm if RAB4, the fast loop recycling GTPase, is the regulator of this internalization of alpha V beta 3, the experimenters then conducted the same experiment using dominant negative mutants of RAB4, RAB11, and ARF6, another GTPase. What they found is that while recycling of alpha V beta 3 with addition of PDGF was not inhibited in the mutant ARF6 and mutant RAB11 additions, the mutant Rab4 constructs do inhibit efficient recycling of alpha V beta 3. This indicates that Rab4 is most likely the main GTPase involved in the alpha V beta 3 pathway. Lindsay, Maya, back to you.
Thank you, Joanna, for the eyewitness report from the site. Oh, looks like Arashiro has some more information for us. So, what do you want to tell us now, Takeshi?
Hello again. In this experiment, recycled cell surface integrin was visualized using biotin tagged antibody with or without the growth factor. J shows the cells without the growth factor where there was not much recycled integrins detected. K here shows the cells with growth factor where many integrin puncta are observed. Now, they tested the effect of integrin recycling on cell adhesion and spreading. Cells were transfected with wild-type or dominant negative Rab proteins and adhered to either vitronectin or fibronectin. The results are not shown here, but the dominant negative Rab4 inhibited the adhesion of transfected cells to vitronectin, but not fibronectin. The spreading of the cells were compromised by dominant negative Rab4 as shown in this figure. This is the wild-type Rab4, and this is the dominant negative Rab4. The transfection with dominant negative Rab11 had no effect on cell adhesion and spreading, indicating that the only direct recycling of integrin from early endosomes to plasma membrane is critical for alpha V beta 3 function.
It's been a crazy morning with all this breaking news. Summing up today's headlines. Alpha V beta 3 integrin rapid recycling is regulated by PDGF in a Rab4 dependent manner. This in turn promotes cell adhesion and spreading. This regulation of Rab4 may occur via activation of PI3 kinases that can regulate GTP exchange on Rab4.
Thus, these data describe to us, for the first time, a mechanism through which a growth factor like PDGF can regulate late integrin recycling, and that this actually has a functional consequence during cell adhesion and spreading. We will keep you guys updated on the ongoing investigation of the involvement of PI3 kinases and PKB-AKT in growth factor regulated alpha V beta 3 recycling.
More on actin and myosin on the other side of this commercial break.
Stay tuned, guys. Keep watching.